IMC-F106C is a ImmTAC. That's a molecule that's a T-cell receptor on one side and a CD3 engager on the other. The T-cell receptor engages peptides from PRAME protein that are digested and presented by tumor cells. On the other side, the CD3 engager brings the T-cell close. This is proof of principle to show efficacy of this drug in a phase 1 setting for patients with cutaneous melanoma, uveal, ovarian cancer, lung cancer, endometrial cancer. So high PRAME expressing cancers. And of course it was a phase 1, so safety and efficacy was important. The safety is great, no significant toxicities. All of the cytokine release syndromes were 1 and 2, very well tolerated, no real toxicities. No one was discontinued for toxicities, no grade 5 toxicities. Then the efficacy portion is important because this platform has been validated by a similar drug that's been approved by the FDA for ocular melanoma, tebentafusp, which targets GP100. This targets PRAME. So the efficacy here, we saw early responses in patients. In the efficacy population, 31 patients, we saw significant response rates in uveal melanoma, ovarian cancer, and cutaneous melanoma. We saw interesting decrements in correlatives of response, like ctDNA decreases in patients, including non-small cell lung cancer that didn't have a formal response, but had greater than 50% decrease in circulating tumor DNA. Interestingly, 80% of those patients that had ctDNA that was followed, so 20 patients, 80% of them had some decrement, and a quarter of them cleared their ctDNA. So an interesting beginning to this drug. It shows a T-cell targeted therapy that's off the shelf, evaluable and utilizable for a whole host of solid tumor types, whole host of patients, age, performance status, etc. And as we expand, we have expansion cohorts in multiple tumor types, and we're moving this into combinations with chemotherapy and checkpoint inhibitors. So stay tuned for more on this interesting drug.